Hey everyone, my name is Indra Krishnamurthy. I am a senior data scientist in Brilio. First of all, thanks everyone for joining us today. Today I'll be talking about AI in drug discovery. This is one field that totally interests me a lot, more so because of the fact that I feel in some way this is a field that touches people's lives in a more direct way, right? Quick disclaimer on your screens. Uh, Before I move on to the agenda, a quick tidbit. This came as a surprise when I first got to know about it, right? Did you know that for a new drug to get approved in the US, it takes almost or as much as 12 to 15 years? It's a whole damn long process. And uh, it's as expensive as it could be as expensive as 2 to 2.5 billion dollars for a single drug, right? What bothers more is the fact that the failure rate is pretty high. So if you look, if you search for the failure rate, it's as much as 90%. And most of the time, this happens halfway from from when they start with the drug development process, somewhere during the clinical trial stage. Right. Having said that, let me quickly move on to the agenda. I plan to start with explaining what is the drug discovery process, what are the challenges. We'll try to understand the different stages within the early drug discovery phase. And then I'll move on to explain a few use cases on how AI can be used in early drug discovery, followed by what are we to look out for when it comes to the adoption, as well as how machine learning or AI is is being looked at by the specialists, right? So what is drug discovery, right? When you are looking at the drug development and approval process, the diagram on your left, uh, we'll find that it starts with the early drug discovery stage. Now, this is a process through which new potential therapeutic entities, new drug components or compounds are identified using a combination of computational, translational, and clinical models. Now, the substances or the compounds that are identified in the early drug discovery stage are refined and extensively tested in a laboratory setup and in animal or alternative models when it comes to the preclinical research. The sole aim here is to provide sufficient proof of safety and efficacy before before the compound is basically tested on human beings in the next stage, that is of the clinical research. Now, the clinical research is one of the most longest processes, which consists of 3 to 4 phases, where different groups of people in increasing numbers, the drug's efficacy and the dosage are determined and tested. Now, post the third phase is the last stage of the drug development process, which is called the regulatory review and approval, where we get the approval from a national regulatory authority for the drug to be consumed by the patients, or to be published in the market. So I will start with understanding the different stages in the early drug discovery stage. Now, in order to, this becomes mandatory to understand the next 4 slides, which is on the application of AI and ML in these processes. So again, there are 2 terminologies which I would like to explain. One is that of target and the other is hit. Now, target is a broader term which is used to refer to a range of biological entities, like say a gene or a protein that plays an important role in a disease. So you need to figure out which is that druggable entity in the body that is accessible to a drug molecule and which has some kind of a biological response upon binding, right? One of the first steps is to identify this target in the body and then validate the target through multiple approaches like, say, animal models again, or bioactive molecules, cell-based models, functional analysis of genes, and so on. Now, once target identification and target validation happens, the next is the assay development and hit discovery process. Now, hit is defined as a molecule or as a compound that will interact with the target. So we're now talking about the drug molecule, which will interact with the target and result in some kind of therapeutic effect, giving some relief. Now, in order to identify hit, there are multiple strategies used. Here is one application which extensively uses robotics, data processing, and other software, right, when it comes to the high throughput screening. There's also phenotypic screening, fragment-based screening, and so on, which are some of the strategies used for hit discovery. Now, once hit discovery, post the hit discovery stage, the hits obtained from the VS or the HTS are triaged to filter out the compounds. One of the ways to identify the or compute the potency of candidate compounds is through the value called IC50, which gives the half maximal inhibitory concentration. This is done through plotting what is called the dose response curves, which looks like a logistic curve. Now, once the compounds, once the hits have been selected, the next step is to group them into clusters to form what are called lead series based on their structure. The next step is to is called the hit to lead or H2L process, where small molecule hits from an HTS are evaluated and optimized into lead compounds, right? That is called the hit to lead step. The final stage is called the lead optimization stage, where the compounds that were discovered in the H2L process are synthesized and they are modified to improve the potency and reduce whatever side effects that could be. Now, this step also conducts experimental testing using animal efficacy models as well as Admet tools, leading to the designing of the drug candidate. So this is about the different stages. Let's just move on to the use cases of where AI is used in some of these processes, right? First use case I wanted to talk about is on the prediction of the ADMET properties, right? This refers to the chemical absorption, distribution, metabolism, excretion, and toxicity. Now, these play a key role in drug discovery. Why so? When I started off this presentation, I was talking about the failure rate of this drug discovery process, right? Now, two of the major causes leading to this is the lack of efficacy and safety, which means since these properties are indicative of efficacy as well as safety, these play a vital role in every stage of drug discovery and development. So, a high quality drug candidate should not only have sufficient efficacy against the disease or the therapeutic target, but also show appropriate ADMET properties at a proper dosage. Now, instead of limiting the analysis of these properties to just a few leads, through the use of machine learning algorithms, it becomes possible to screen thousands of compounds very quickly. Right. So we had used a similar approach for bioactivity prediction for IC50 value prediction. Let me quickly talk about the process, right? On the left-hand side, you have the chemical structure of the input molecule. Now, these molecules can be encoded by a string of text known as the SMILES notation, which looks like this. Now, these SMILES encoded molecules can then be converted into what is called molecular descriptors using a package like RDKit. Now, what are these molecular descriptors? These are information about physical and chemical properties of the molecule. Now, for example, a 1D descriptor could be talking about the count or the type of atoms in the molecule, or it could be the molecular weight, whereas a 2D descriptor could talk about the size and shape, and more complex stuff like the electronic distribution and so on. Now, once we have these uh, values, right, they're basically like columnar values in your data frame, and then you could build multiple regression models to uh, figure out, say, for example, the IC50 value or the um, absorption or toxicity level and so on, right? So this was the first use case I wanted to discuss about. The second use case is to predict the drug target interaction using machine learning algorithms. Now, this can help in screening out unsuitable molecules on one hand, and on the other hand, only a small number of proteins have been identified as active and are targets for currently approved drugs, which means that the pharmacological functions of most proteins are not yet are not yet known for most compounds, right? So in this particular implementation, the input is a drug target pair, where the drug uses the SMILES notation and the target uses the amino acid sequence. The output is a classification model which indicates the binding activity of the drug target pair. It could as well be treated as a regression model, which could predict the affinity model and give a, I mean, which could also predict the affinity score, right? There are different perspectives when it comes to the way the feature engineering is done. A lot of times, CNNs and RNNs have been used for generating for doing the feature engineering step, which is then fed to a model. It could again be a fully connected layer or doing the prediction, or it could be an SVM model or a random forest model, which can be used for predicting the interaction, right? The third use case is how we could use the enormous amount of a sequence or text data available in the biomedical world. Some of the examples could be biomedical named entity recognition, relation extraction, assertion models, summarization that can basically literally accelerate target identification, or even different stages in the different other stages in the drug design. We got a chance to work with clinical protocol documents, which lets us uncover a lot of information in terms of the entities and the relationships. A few examples are on your screen where multiple NLP tasks have been listed down across the data sets and the algorithms used. The first one is a named entity recognition model called BioBot. The second one is more about extracting gene-disease relationship from documents, which uses a CNN and a attention-based BiLSTM model. We also see how a text summarization algorithm has been built over the PubMed dataset and so on. The final use case that I wanted to talk about today is how AI is used in drug repurposing. So it's also called target extension. This allows the prediction of new potential applications of medications, right? In this particular implementation, they had used gene expression data or transcriptional profiles alone. Now, we're talking about data sets that are huge, high dimensional, as well as very complex and sparse information. For me, the highlights of this paper was how the feature engineering and dimensionality reduction was applied. So in order to preserve the biological information as well as allow for reduction at the same time. Now, there are two methods that were described here. One is the pathway activation scoring, and then gene expression for landmark genes. So in the first one, all the samples were analyzed, and for each sample, they calculated what is called the pathway activation profile for 271 signaling pathways. And in the second case, in the second approach, they used a data set containing normalized gene expression for 977 genes. From the Links project, the authors claim that almost 80% of the biological function was captured, and it had a great inferential value. So these are some ways where machine learning and deep learning have been applied to drug discovery process. This is just few ways. These are just few papers that I had gone through. This is, like I said, very interesting, and it is a very domain heavy field. The adoption is still not as great as it has been in other industries, right? It is still received with some kind of skepticism. Now, a couple of factors that might be leading to these challenges is probably the very nature of biology and chemistry, where experts say that AI still needs to grow. Our algorithms still need to grow in a more sophisticated manner to understand the context that comes with this field, due to which the trust factor is still missing when it comes to AI in drug discovery. After all, it deals with human lives, right? The black box nature of the AI systems, which we're always questioned about. But thanks to explainability AI and also the steps or the research that's being done in order to make our algorithms more explainable, probably the adoption of AI along with getting the best out of these systems, I feel would require more interdisciplinary teams working together with a feedback loop, human in the loop kind of systems, which will make our algorithms learn better and hopefully in course of time improve the trust on artificial intelligence in this field. Thank you so much for joining me in this. I hope this was of use to you. Thank you so much once again for giving me this opportunity. Quickly before I say a bye, these are the papers that I had referred to. Thank you so much. Have a great day.